Welcome to the Explainer. So today we're diving into one of the absolute most elegant, highly coordinated systems known to science. We usually picture the human body as this giant machine, right? But honestly, it's way more accurate to look at it as an economy, a biological economy that meticulously manages its currency. And in this economy, the primary currency is energy. So let's explore exactly how the body manages its biological bank accounts because, let me tell you, it completely changes how you view your own metabolism. Now, I've got a question for you. How exactly does your body keep running when the literal food runs out? I mean, think about it. We face this massive logistical problem every single day. We eat meals hours apart, but our brains and our muscles, they demand a constant, uninterrupted supply of energy. No breaks. So when the food runs out, your body's internal enzymes have to step up. These guys are the ultimate problem solvers, making split-second decisions just to keep you powered.
Okay, let's dive right into our roadmap for today. We're going to cover making glucose from scratch, the teamwork of the Cori cycle, glycogen as our savings account, how we control that energy supply, and finally, what happens when the whole system fails.
Kicking things off with Section 1, making glucose from scratch. So, when our biological cash reserves start running dangerously low, we literally have to manufacture more. And that brings us to a really foundational concept, gluconeogenesis. Simply put, this is the synthesis of glucose from non-carbohydrate sources, stuff like pyruvate or lactate. Your body is basically running the metabolic machinery in reverse. But, your cells are smart. This process isn't just running all the time willy-nilly. It's favored specifically when the cell is already meeting its current energy needs and has a healthy stockpile of specific molecules, like acetyl-CoA or citrate.
And the absolutely crucial point here is reciprocal regulation. The body essentially prevents glycolysis, which is breaking down glucose, and gluconeogenesis, which builds it, from happening at the exact same time. It's like trying to build a brick house while someone else is simultaneously tearing down the walls. Total waste of energy, right? So the body uses these clever metabolic control points. For instance, when a molecule called AMP rises, it acts as this incredibly sensitive alarm bell. Seriously, it's way more sensitive than ATP. It signals that the cell is practically starving for energy, which then favors glycolysis. Conversely, a spike in citrate or acetyl-CoA, that tells the cell, hey, energy is plentiful, giving the green light to gluconeogenesis.
Moving right along to Section 2, the Cori cycle teamwork.
Okay, so I just mentioned that building and breaking down glucose can't happen in the exact same spot. But get this, they can happen in completely different organs at the exact same time. And this brilliantly illustrates the Cori cycle. During a heavy workout, your metabolically selfish muscles are working incredibly hard, breaking down glucose into lactate. Muscles literally hoard glucose strictly for their own contraction. They do not share. So they dump all this leftover lactate straight into the bloodstream. Enter the liver. Acting as the generous central bank of the body, the liver scoops up that lactate, uses gluconeogenesis to turn it back into fresh glucose, and ships it right back out to the muscles. It's just a perfectly designed physiological loop.
And I always love to highlight the human element behind these major breakthroughs. This cycle is actually named after Carl and Gertie Cori, who took home the 1929 Nobel Prize for figuring out this exact metabolic link between the muscle and the liver. Honestly, almost 100 years later, it remains an absolute cornerstone of modern biochemistry.
All right, shifting gears to Section 3, glycogen, our biological savings account. Let's take a look at how we actually store all this energy. What we're looking at is the physical structure of glycogen, literally our biological savings account. It's this highly branched polymer of glucose. Notice how it only has one single reducing end at the core, but it has all these multiple non-reducing ends on the outer edges of the branches. Now, why does that physical shape even matter? Well, having all those multiple non-reducing ends allows for lightning-fast glucose mobilization. It's exactly like a bank having 20 tellers open at the same time instead of just one. When you need cash quickly, like say you're suddenly sprinting away from danger, enzymes can simultaneously withdraw glucose molecules from every single one of those outer ends at once.
So, how do we actually break the bank? It's a three-step mechanical process called glycogenolysis. First up, an enzyme called glycogen phosphorylase starts cleaving off glucose units. It's what we call a processive enzyme, meaning it grabs on and just enthusiastically snips its way down the line. But it eventually hits a roadblock when it gets within 4 glucose units of a branch point.
That's when player number 2, the aptly named debranching enzyme, steps in. It transfers that whole branch out of the way, which finally allows that last stubborn branch in glucose to be cleaved off.
And as we see here, this is that sequential removal in action. That glycogen phosphorylase enzyme uses a specific little cofactor called PLP to literally snip off the terminal glucose residues, converting them into glucose 1-phosphate molecules. It just relentlessly marches down the chain, freeing up energy bit by bit until it slams into that structural roadblock we just talked about.
Now you might be wondering, how do you even open an account like this in the first place? Because the enzyme that builds glycogen actually cannot start a chain from scratch. It desperately needs a primer. Enter glycogen. This is the core protein sitting at the very center of the glycogen particle, acting as the ultimate anchor. It jumpstarts the whole operation, allowing the body to build up this massive biological savings account of up to 50,000 glucose molecules, all surrounding this one single central protein. Isn't that wild?
Next up is Section 4, Controlling the Energy Supply.
With a massive savings account like that in place, we need some pretty strict managers to make sure the bank isn't accidentally depositing and withdrawing funds at the exact same time. This control relies heavily on the physical shape or conformation of our key enzyme glycogen phosphorylase. It basically exists in two distinct states. You've got the R state, which is the active phosphorylated state. Think of this as the withdraw mode. And then there's the T state, the inactive unphosphorylated state. That's the save mode. By shifting the enzyme back and forth between these two shapes, hormones and cellular signals can expertly pull the levers of your entire internal economy.
Now, what's really interesting about this is how fast it happens in your muscles. Let's say you suddenly start sprinting. That intense energy demand causes a massive, immediate spike in AMP inside your muscle cells. Without even waiting for a hormone to come along and tell it what to do, that AMP directly binds to the enzyme and forces it right into the active R state. This allosteric control triggers energy release in mere minutes, way before the hormonal epinephrine signal, the adrenaline rush, even arrives from the bloodstream.
But let's bounce over to the liver because it paints a completely different picture. Remember, the liver isn't selfish like the muscle. Its job is to regulate blood sugar for the entire body. Here, if there's high blood glucose, that glucose directly binds to the phosphorylated enzyme, and boom, it immediately forces the enzyme into the inactive T state. It's this brilliant rapid response mechanism that literally shuts off glycogen degradation the second glucose is plentiful. It stops the liver from over-withdrawing before the hormone insulin even has a chance to kick in.
Speaking of hormones, they are the undisputed bosses of this whole economy. Glucagon and epinephrine, they act as the withdraw signals, promoting degradation. Insulin, on the other hand, is the deposit signal, promoting synthesis. And here's a totally fascinating specific detail for you. Muscle cells completely lack glucagon receptors. Remember what I said earlier. They are metabolically selfish. They couldn't care less if the rest of the body's blood sugar is tanking, so they completely ignore glucagon. They strictly respond to epinephrine and only when it's absolutely time for that fight or flight energy.
Finally, let's wrap up with Section 5, when the system fails.
So what actually happens when a piece of this incredibly tuned machinery breaks down? Well, we see the real-world consequences when we look at glycogen storage diseases. Notice how a defect in just one highly specific enzyme causes totally different, devastating issues depending on where it happens. If you lack the glucose 6-phosphatase enzyme in the liver, you get von Gierke disease, leading to severe hypoglycemia and kidney failure simply because the liver can't release glucose into the blood. But if you lack muscle phosphorylase, you get McArdle disease, resulting in painful exercise-induced muscle cramps because the muscle can't access its own local energy stores. Just one single missing enzyme, but it leads to completely different systemic catastrophes.
Which leaves us with this final, really striking thought. When a single microscopic enzyme in this economy declares bankruptcy, the entire biological system cascades into failure. Honestly, it makes you marvel at the fragile yet highly coordinated perfection operating flawlessly inside of you every single second. It makes you wonder what other hidden biological economies are secretly running inside you right now. Keep feeding that curiosity, and thanks so much for exploring this with me today.